So let's straight away start with our topic. So I hope that you must have revised the previous topics. So in the last class, we have studied some of the cell organelles in the eukaryotic cell. Mainly we have talked about the cell membrane and cell wall. Now today we are going to continue with the same section, and today we are going to start with the new cell organelle that is called endomembrane system. Endomembrane system, as you can see here. Now here this endomembrane system, why is it called system? Anything which is called system has more than one component, that's why it is called system. So here endomembrane system will consist of some cell organelle, like it will consist of endoplasmic reticulum, also called as ER. It also consists of Golgi bodies. It also consists of Golgi bodies. It consists of lysosome. It consists of vacuoles. So this first thing you remember. If I say wherever it is written that endomembrane system consists of, so your answer should be it consists of these four components. Now if I ask you, is cell membrane a part of endomembrane system? No. Mitochondria part of endomembrane system? No. Peroxisome? No. Chloroplast? No. Right? Only these four are the part of endomembrane system. Then the question comes, why only these four are the part of? Because they are structurally and functionally interrelated. They are structurally and functionally interrelated. Now in NCERT, if you read, there is one more point which is given clearly, and they have said that, and it is asked in previous year NEET also, that whether mitochondria, chloroplast and peroxisome are they a part of endomembrane system? So they are not the part. They are not a part of endomembrane system. They are not the part of endomembrane system. Now, when I say structurally and functionally interrelated, then here is this, just see the interrelation over here. See, this part is representing the ER. This is endoplasmic reticulum or ER. This part is the Golgi bodies. This part is the lysosome. And there is, let's say, vacuoles here. Now, if you see, then you see the arrow. The vesicle coming from the endoplasmic reticulum is entering inside the Golgi. Now from Golgi you can see lysosome is coming out, and from Golgi only this vacuole is also coming out. So if you see, the lysosome, vacuole are formed from Golgi body, and Golgi body is continuously connected with the ER, right? So now here if we see, ER is connected with Golgi body, is connected with lysosome, and then this is also connected with vacuole. So I hope that you all understood endomembrane system are interconnected. ER, Golgi, lysosome and vacuole. Now next here, if this is the question, I expect all of you to answer this question. Now I am waiting. Let me see who will answer first. Yes, let me see who will answer first. The endomembrane system in eukaryotic cell include all of the following except, all of the following except, yes, all of the following except. Which one will be the right answer? All of the following. I am waiting. Yes, Khalil answering D, Chetna D, Ravi Kumar C, okay, Karthik D. So see, clearly I have mentioned that mitochondria, chloroplast and peroxisome are not a part of endomembrane system. So mitochondria is not a part of endomembrane system. ER, Golgi and nucleus, they are part of endomembrane system. Endomembrane system. So is it clear? So now let's move with the next topic. So Matant, you also answer the question. Answer the question. Answer. Now here ER. So if we see ER here, endoplasmic reticulum is of two types. First one is rough endoplasmic reticulum and second one is smooth endoplasmic reticulum. If we see here clearly, if this is nucleus, so outermost layer of nucleus, understood? The outermost layer of nucleus is connected. Let's say this is the outermost layer. So it is connected with the endoplasmic reticulum. It is continuous with the endoplasmic reticulum. And here you can see ribosome is present on the surface. So first of all, ER is divided into rough endoplasmic reticulum and smooth endoplasmic reticulum. In short, we call it RER and ACR. RER and ACR. So rough endoplasmic reticulum has ribosome present on the surface. Ribosome present on the surface. When the ribosome is present, then it will give a rough appearance. Now, when ribosome is present, then what will be the, if ribosome is present, what will be its function? So its function will be protein formation. Its function will be protein formation. Then ACR, they lack ribosome. When they lack ribosome, so their surface will be smooth or rough. So their surface will be smooth. When their surface is smooth, then that's why it is called smooth endoplasmic reticulum. And what is its function? Its function is lipid synthesis and storage. Lipid synthesis and storage. Lipid synthesis and storage. So understood this? Now here one more function of ACR is it help in steroid hormone. A steroid hormone synthesis in animals. Clear? So ACR lack ribosome. They are smooth because they lack ribosome, and the lipid synthesis, storage and the steroid hormone formation in animal. So is this point clear to all? So now difference, if here in exam question is asked, how can you identify RER and ACR? So how will you identify? If ribosome is present on the surface, then it is, so as you can see, this is RER and this one is smooth. Ribosome is not present, so it is SER. Is it clear? Next. So this is the question. Solve. All of you solve this question. Let me see who answered this question first. Answer. You have 20 seconds. So you have 20 seconds. Answer it. Unmute. So it is okay, correct. So I can see answer is coming B. Okay, fine here. So what is the main function of the rough endoplasmic reticulum? So the main function is protein synthesis and modification, because rough endoplasmic reticulum have ribosome. Rough endoplasmic reticulum have ribosomes, and they help in protein synthesis and modification. Remember this point, protein synthesis and modification. Next question. Now, this is regarding the smooth endoplasmic reticulum. Smooth endoplasmic reticulum. So, smooth endoplasmic reticulum are involved in fourth. What will be the answer here? Yes, I am waiting. Smooth endoplasmic reticulum. Okay, Bhargav said B, Punya B, Karthik B. Okay, B, Nikhil B, Khalil B. Very good. So all of you are answering correctly. So the smooth endoplasmic reticulum is involved in lipid synthesis and detoxification. See, a new term is there, detoxification. So from here you now have three functions of ACR. One function is lipid synthesis. Lipid synthesis. First function lipid synthesis. Second function steroid hormone synthesis. And third function is detoxification. Detoxification. Now, if I ask you that steroid hormone synthesis is formed in plant cell, true or false? Not in plant cell, it is in animal cell. Remember, this steroid hormone in animal cell. This point is clear. Now, let's move ahead. So the next cell organelle in endomembrane system is Golgi bodies. Now Golgi bodies, as you can see here the structure, you can see Golgi body has three types of structure. The circular structure, as you are seeing here, the circular structure is called vesicle. The circular structure is called vesicle. This flat membranous structure, as you can see, these flat membranous structure are called, they are called cisternae. These are called cisternae. And then there are small tubule like structure. So what you can say, Golgi body can have cisternae, tubules and vesicles. Since they have cisternae, tubules and vesicles, so they are used in, since they have cisternae, tubule and vesicle, so they are used, or they are called as pleomorphic. They are called as pleomorphic cell organelle. Remember, question if it is asked, which cell organelle is pleomorphic in nature? Which cell organelle is pleomorphic in nature? So you will say it is the Golgi, because some of the membrane are in the form of cisternae, some are in the form of vesicle, and some are in the form of tubules. Now, next coming to this, further if you see it, Can we say that cisternae are parallel to each other? Several cisternae are there and all of them are to some extent parallel to each other. Further, if you see, they have two types of face. They have two types of face. One face you can see cis face. Another face you can say trans face. So they have trans and they have cis face. So they have two faces. They have two faces. They have two faces. Now the cis face is called incoming phase. The cis phase is called incoming phase. Incoming phase. And the trans phase is called outgoing phase. Trans phase is called outgoing phase. Clear? Now these two phases are there is a line of NCERT. I am repeating it. Now your task is to go and underline this line, underline and find out this line in NCERT. It says that the two phase of Golgi bodies are different but they are interconnected, right? These two phase around this in NCERT it is given the phases are different. The phases are different but they are interconnected. Different but interconnected. Is this point clear? They are different but interconnected. So this is all about the Golgi body. So this is about the structure only. Now we will see the function of Golgi body. What is the main function? So if we see the function here, remember there are three main functions. The first function is, see here cis phase I have said incoming phase, and incoming means here I have shown that it is coming from where? Endoplasmic reticulum. The vesicle is coming from endoplasmic reticulum. Now vesicle is coming from smooth also and rough also. So from rough endoplasmic reticulum protein will come and smooth endoplasmic reticulum lipid will come, right? So here what will happen? In first will be modification. Modification of what? Modification of protein and lipid coming from ER. So here protein will become glycoprotein and lipid will become glycolipid. Lipid will become glycolipid. So if question is asked, glycoprotein and glycolipid is formed in, what will be your answer? Glycoprotein and glycolipid is formed in. So your answer should be, it forms in Golgi bodies. Further, let's say, further if the question is asked, now another point where, so first function is once it is modified, then it will be packed inside a vesicle and it will be sent into the different part. So first is modification, second one is packaging. So packaging, what do you mean by packaging? Packaging in vesicles. Packaging in vesicles. And third function is secretion. Secretion means both intra and extracellular secretion. Now, let me explain this point a bit. All three function I will explain. So all of you pay attention here. From here question is asked. Now, if I say function of Golgi body, so the first function of Golgi body is modification. Modification of protein and lipid. protein and lipid modify and become glycoprotein and glycolipid. Fine. Second function, these glycoprotein and glycolipid will be packed inside a vesicle. So this is called packaging. Once it is packed inside the vesicle, then it is secreted. Now this secretion will be intracellular, extracellular, or both? So the answer is both. The secretion will be intra and extracellular both. Also additional, we have already seen that the lysosome formation is also the function of Golgi body. If I ask you this question, which cell organelle help in the formation of lysosome? You will say it is Golgi body. So now let's move ahead. So I think this point is clear. Now if we see here, so all of you, question time, get ready to answer. Question time, get ready to answer. Here is the question. Which organelle is responsible for modifying, shorting, packaging protein for secretion or delivery to other organelle? Simple question. Venkata has given answer. Venkata has already given answer. Very good. Amruta Lakshmi given answer. Shravani, Ganesh, good. Lakshmi, Faz, Lashya, Khalil, Ganesh, Nikhil, very good. So you all are answering Golgi apparatus. Yes, correct. It is Golgi apparatus. So we have seen that the function is modification. Shorting means packaging, right? Shorting, packaging, and also secretion. So now because of its function, this is also called as the middleman of the cell. Golgi body is also called as middleman of the cell. You can note it down. Middleman of the cell. It is sometimes also called as the postman of the cell. Postman of the cell. It is sometimes also called as the traffic police of the cell. Traffic police of the cell. Now see, either call it middleman of the cell, call it postman of the cell, or call it traffic police of the cell. It's the same thing. All are used for what? All are used for Golgi bodies. So these names you should keep it in mind. Now moving ahead. Next cell organelle is lysosome. So lysosome, as I explained, that it is formed from Golgi. It is formed from Golgi bodies. Now here lysosome, inside the lysosome if you see, there are hydrolytic enzyme. What is this hydrolytic enzyme? Hydrolytic enzyme, basically they will have a group of enzyme which include protease. Now see, these all enzyme may be new for you. So if you find them new, just note it down. What is the function? Protease. Protease will digest protein. Protease digest protein. Lipase. Lipase will digest lipid. Lipase will digest lipid. Then comes amylase. Amylase digest carbohydrate. Carbohydrate. And there is another group of enzyme, nuclease. It digests DNA and RNA. Now if I say in cell, lipid, protein, carbohydrate, DNA, RNA, only these are present. If hydrolytic enzyme can digest everything, so do this lysozyme has the ability to digest the entire cell? Answer is yes. It has the ability to digest the entire cell. Entire cell. That is why it is also called as the, it is also called of, you must have heard, suicidal bag of the cell. It is also called as the suicidal bag of the cell. Suicidal bag of the cell is lysosome. Now, one more important thing about lysosome is that here the hydrolytic enzyme is present, and these hydrolytic enzyme require acidic pH. Acidic pH. All of you tell me what pH is called acidic pH. Write in the comment box. More than 7, less than 7 or 7. Which is acidic pH? Is it less than 7 acidic or more than 7 acidic or 7 is acidic? Yes, quickly answer. What is an acidic pH? Acidic pH is, very good, Khalil has given answer, less than 7. Khalil has given, okay, Kalyani also said less than 7. Vashu, less than 7, fine. So now I am getting all of you as less than 7, very good. So less than 7, so acidic pH here is approximately 5. So the pH here is approximately 5, that's why we call it acidic pH. Now the question is, why acidic pH is required? Why acidic pH is required? So acidic pH is required because hydrolytic enzyme are active only at acidic pH. Right? The hydrolytic enzyme are only active at acidic pH, that's why an acidic pH is required. Now, if I say that how many membrane is surrounding the lysosome, is it single membrane bound cell organelle or double membrane bound cell organelle? So it is a single membrane bound cell organelle. Single membrane. Another important point, we have seen it is called as the suicidal bag of the cell. Is there any other name for the cell? Yes, there is one more cell. It is also known as the cleaner of the cell. You also can call it the cleaner of the cell. It is the cleaner of the cell. It is the cleaner of the cell. Understood this point? So who is the cleaner of the cell? Lysozyme. Who is the suicidal bag of the cell? Lysozyme. Suicidal bag of the cell has what kind of enzyme? Hydrolytic enzyme. Hydrolytic enzyme are active at which pH? Acidic pH. What is the pH? 5. Why acidic pH? Because the hydrolytic enzyme are active at acidic pH. What are the component of hydrolytic enzyme? Protease, lipase, amylase, nuclease. They will digest protein, lipid, carbohydrate, DNA and RNA. Since they have the ability to digest all, they are called the suicidal bag of the cell. So this is about the lysosome. Now question related to lysosome. I am waiting for the answer. Lysosome are responsible for, are they responsible for protein synthesis? Are they responsible for synthesis of lipid, modifying protein? Sujata is answering B. Very good. Chandrika B. Lashya B. Very good. Reshma B. Yadav B. Fine. So all of you are answering B. It means point is clear to all of you. Yes, point is clear to all of you that lysosome are responsible for breakdown of waste material and cellular debris. They are responsible for breakdown of waste material and cellular debris. Clear? breakdown of waste material and cellular debris, and that is why it is called cleaner of the cell. Right? It is also known as, another name is housekeeper of the cell. Housekeeping means cleaning, right? So it is also known as housekeeper of the cell. So these are some of the name given to lysosome. This point very much clear. Now, so let's move on to the next point. Next cell organelle is vacuole. So vacuole is the fourth part of the endomembrane system. So here if we see, it is also surrounded by a single membrane, as we can see here. Now the membrane of vacuole is called tonoplast. Remember, again previous year NEET question it is asked. The membrane of vacuole is called, so it is called tonoplast. It is called tonoplast. Then inside is the cell sap. Vacuole is membrane bound. It is formed from, this is also formed from, Golgi bodies. It is also formed from Golgi bodies. Clear? It is also formed from Golgi bodies. This vacuole has a very important role, right? Although they don't have a very general role in all the cells, just like other cell organelle, they have different role in different cells. So here we will summarize all the role. So main common role is they are used for storage. They are used for storage. They may also used for osmoregulation and excretion. Osmoregulation and excretion in which cell? In amoeba. Amoeba or paramecium. There they will help, and this vacuole which has a role in osmoregulation and excretion is called contractile vacuole. Contractile vacuole. So you can say contractile vacuole is a type of vacuole which has a role in osmoregulation and excretion, and it is found in amoeba and paramecium. Another type of vacuole is called food vacuole. Food vacuole. Now it has the role in digestion of food. It has a role in digestion of food. It has a role in digestion and storage also. This is also found in amoeba. So now remember, if I ask you how many types of vacuole are there, so you will say two types of vacuole, contractile vacuole and food vacuole. Where is contractile vacuole found? Amoeba and paramecium. Where is food vacuole found? Again in amoeba and paramecium, but paramecium have complex mechanism for food, so mainly you can say amoeba. Now if you can recall, in bacteria we have studied about gas vacuole. So is gas vacuole will be considered as a vacuole, true or false? If I say gas vacuole is a true vacuole, true or false? Write down. Tell me, gas vacuole is a true vacuole or not? Gas vacuole, it is not a true vacuole. Gas vacuole is not a true vacuole. That's why we will not cover that part here. It is only present in prokaryotes. It is only present in prokaryotes. One more important point. Very good. Very good. Most of you are writing false. All of you are writing false. Very good. Priya, Reddy, Mahi, Utkarsh, Honey. Very good. So false. So now one more important point. This vacuole form note this point, very important, it form 90% of volume of cell in matured plant. In matured plant cell. Matured plant cell. Is it clear? So it occupy 90% of the volume of cell in matured plant cell. Now one question again. If I ask you again, this is a true-false question. Answer it. I am writing it here. If I say vacuole is present in animal cell, true or false? Vacuole is present in animal cell, true or false? Vacuole is present in animal cell, true or false? Vacuole is present in animal cell, true or false? Yes. So answer is false. True. Okay. True. False. So true false mix is coming. See, remember, vacuole is present in both plant cell as well as animal cell. Vacuole, repeat with me, vacuole is present in both plant cell and animal cell. Vacuole is present in both plant cell and animal cell. Here, but difference is in animal cell the vacuole is small in size. Vacuole is small in size, whereas in plant cell vacuole is large in size. In plant cell vacuole number is only 1, but in animal cell the number will be multiple. So I am drafting the sentence. Listen carefully. In animal cell the vacuoles are present, but number of vacuole is large and the size is small. Whereas in plant cell the vacuole is present and the size is very large and number is only 1. So this is the difference between vacuole of animal and plant cell. Now next, mitochondria. So next cell organelle is mitochondria, very important cell organelle if we see here. So if you see here this mitochondria, as you can see, there is an outer mitochondrial membrane, as you can see here. There is inner mitochondrial membrane. In between the outer and inner mitochondrial membrane, there is a intermembrane space present, right? There is an intermembrane space. See, I will draw one diagram. See, this is the outer membrane. This is the outer membrane. Then there is an inner membrane. Now, inner membrane is having several infolding, as you can see here. It has several infolding present, right? Outer membrane, there is inner membrane, then inside is the matrix. This in between them is the intermembrane space. Intermembrane space, right? Now, this inner membrane is folding and this fold is called, as you can see here, it is called cristae. It is the finger-like projection. The inner membrane is folded and it forms small finger-like projection called cristae. Cristae increase the surface area. Now, inside you can see there are several ribosomes present. So, ribosomes is also present. Then, apart from this, they can also have circular DNA called plasmid. They have circular DNA called plasmid. So this is overall the structure of mitochondria. Now, what is the main role of mitochondria? So, they help in aerobic respiration and they help in production of ATP. So, write it down. They have a function in aerobic respiration. And since they help in production of ATP and energy, so that is why they are called the powerhouse of the cell. They are called powerhouse of the cell. So if asked which cell organelle is called the powerhouse of the cell, so it is mitochondria. Mitochondria is helpful in aerobic respiration or anaerobic respiration. So it is aerobic respiration. So I hope this point is clear. Mitochondria. Now here later on in respiration, in aerobic respiration, we will see that here in matrix some reaction will take place, in the inner membrane some reaction will take place. That we will see in the respiration chapter. So I think this point is very much clear to all. Now, because it has its own ribosome and it has its own DNA, so this cell organelle is called semi-autonomous. Remember this point. Which cell organelle is called semi-autonomous? So it is mitochondria, because it has its own ribosome and it has its own DNA, right? Now, one more thing. How do this undergo division? So they divide. They divide by fission. They divide by binary fission, just like prokaryotes. They divide by fission. So overall, these are the points about the mitochondria. Now, question about mitochondria. All of you solve this question. So the question is, first one, what is the primary function of mitochondria? What is the primary function of mitochondria? All of you who are connected, if you are enjoying the video, if you are understanding the concept, do hit the like button, because only 22 of you have liked the video. Yeah, and answer quick, fast. What is the primary function of mitochondria? What is the, very good, Tanish, correct, C, Reshma, C, Ganesh, C, Khalil, C, Lakshmi, C, Jha, Reddy, Reshma, Karthik, Ganesh, Nikhil, very good, well done. So all of you are answering nicely. So here, what is the primary function of mitochondria? So primary function of mitochondria is energy production. Energy production is the primary function. That is why it is called as the, since energy production is the primary function, that's why it is called as the powerhouse of the cell. Powerhouse of the cell. Next question. Mitochondria contain their own DNA. What does this suggest about their origin? Now, it's a little tricky part. Just think and answer. Think and answer. Mitochondria contain their own DNA. What does it suggest about their origin? So, okay, so I am seeing different answers. D, C, D, D. Many answer is D. Why is D answer? No, D is not the right answer. D is not the right answer. Mitochondria contain their own DNA. What does this suggest about their origin? Formed from rough endoplasmic reticulum. Is it? Mitochondria is not formed from rough endoplasmic reticulum. It is Golgi which is connected to rough endoplasmic reticulum, and no role of rough endoplasmic reticulum with DNA. So D is not the answer. Still many of you are writing D. Okay, B, C, A. So all possible answer you are giving. So let's see this one by one. Mitochondria contain their own DNA, right? Yes, they have their own DNA because they have a circular DNA called plasmid. They have a plasmid which is the circular DNA. So this is correct. Circular DNA. Now, so do they originate from the nucleus? No. Circular DNA, they do not originate from nucleus. They evolve from bacteria through endosymbiosis. Now, let's understand this point. Evolved from bacteria from, do bacteria have plasmid? We have just studied. Do bacteria have plasmid? Yes or no? Bacteria have plasmid. Yes, bacteria have plasmid. So one point. Bacteria also have divide by fission, and the size of mitochondria is also same as bacteria. So all the points are indicating that they are very much similar to bacteria. Size is similar to bacteria. They divide by binary fission, and they have plasmid and ribosome both like mitochondria. So that is why they evolved from bacteria through endosymbiosis. Remember, this term is very important. Endosymbiosis. Endosymbiosis means, remember, mitochondria is formed through endosymbiosis. Mitochondria is formed through endosymbiosis. So this point is clear. Let's move on to the next question. Now, next question, the inner membrane of mitochondria is folded into structure called, inner membrane is folded into finger-like projection. What is that called? What is that called? Finger-like projection. Waiting for the answer. A, B, C, D. Finger-like projection. Remember, outer membrane, inner membrane, finger-like projection. So what this finger-like projection called? So the finger-like projection is called cristae, right? The finger-like projection is called cristae. So correct answer is B. Very good. Nikhil, Karthik, Tanish, Pooja. Very good. So correct answer is B. Then next question. The process of ATP production in mitochondria occur through the process called. Now this is tricky question. As I said, in respiration mitochondria has role, but let me see how many of you can answer it. Fourth, D. Yashashvi is answering D. Okay, B. Tanish, B. Okay. See, process of ATP production in mitochondria occur through a process known as, see, in mitochondria the ATP production take place through, right? So here we will say it is through the citric acid cycle and oxidative phosphorylation. Remember this. Here Krebs cycle and ETS, they both are related to ATP production, and these both take place in mitochondria. And they both are related to ATP production. In detail we will see about this in the chapter respiration. For now, just remember that wherever you see Krebs cycle, ETS, you simply assume that it is related to ATP production and it is taking place in mitochondria. So that is it for today. We will stop here till mitochondria. Chloroplast we will do it in the next class. So I hope you all understood this entire topic. Go back, read NCERT, revise the topic, solve some of the question, again repeat and solve these questions also. So thank you all of you. Have a very nice day ahead.